where he managed daily operations and maintenance of sophisticated flow cytometry instruments. Sir began his career at Rajiv Gandhi Center for Biotechnology, where he supported routine imaging and sample analysis for research scholars, handling advanced microscopy and cell sorting instruments. Very good evening, sir. We are very delighted to have you on board today.
Thank you for the wonderful introduction. And I would like to thank Karnia University for this opportunity, and Jibu sir as well as Murugan sir for this chance to speak with everyone and share my experience with one and all. So I hope everyone can see my screen.
Yes, sir.
Right. So the topic for today is high throughput screening and imaging solutions in clinical research. So my primary aim would be to leave with you guys some technical terms and the brands or the companies that provide these solutions to you, to the various research centers, and give you an overview of what it is or how it is used. I will not be probably going deep into the technicalities, but however, once I will be sharing this slide, and I'll be leaving the web links for the resource pages for each of the technologies. Um, it would be really good if people take note of the companies that I am mentioning, because all of these companies are there in India and they have their direct headquarters in India, which these would definitely serve as a really good opportunities, for the graduates once they get experienced in some of the skills or some of the instrument usages. These are really good places where we can find opportunities for biotechnologies. So to start with, we'll just look at some basics, because most of the cell biology applications where you go into analysis, cell analysis, is based on this particular what to say the I mean the underlying principle is would be this one. So every cell has some proteins or antigens on its surface. So these cell surface markers, as well as there are intracellular markers, based on, so every healthy cell will have a set of markers. These are you may call them CD markers. There are around 300 to 400 CD markers in humans. So each CD marker has a specific function, and companies like BD or Beckman Coulter provide antibodies against these CD markers. So these antibodies are used to detect the various markers. So if a healthy person would have a set of CD markers, and if the person is unhealthy, or if he is having say cancer or leukemia, lymphoma, or any underlying symptoms, some markers may not be present, and some markers that are not present on with healthy people will appear. So that's how you do the diagnosis. So so suppose this is a cell. You have an antibody, which is this Y-shaped molecule, and you have a fluorescent molecule which is attached to the antibody, which enables us to detect the particular cell. Now, how do we detect this cell is, um, each cell goes one by one, hits the laser, and it gives out an emission. And these emissions are detected by various detectors. And that's how we come to know which marker is present in a cell or not. So these markers would tell you which markers are there, as well as another information that we get is about the size of the cell. So we get to know the relative size, how complex the cell is, because each cell, from one cell to another, you have various complexities and granularities. then you get to know the fluorescence intensity of the cell. So these are the basic principles when it comes to cell analysis in research or in clinical sites. This is how we basic stuffs that we look into is the size, granularity, complexity, and the fluorescence intensity. So based on how more a particular marker is expressed, the fluorescence intensity will increase or decrease. Now, when it comes to cell analysis, one of the most commonly used instrument is a flow cytometer, which happens to be my area of expertise. And so I have been involved or have been supporting flow cytometry for almost for 10 years, right from 2013 onwards. So, so flow cytometers comes in various form factors. You have a clinical flow cytometer, research flow cytometer, or you have a flow cytometer that can sort cells into various tubes. So these are the conventional flow cytometers. You will find it. It's very common in most hospitals or research centers. Typical a normal flow cytometer can detect up to like say 13 to 20 markers in a tube at a time. You cannot go more than that. So when it comes to a high parameter flow cytometry, we have a technology called, I would like to introduce something called as CyTOF. CyTOF is cytometry by time of flight. So for a conventional flow cytometry was a normal cell analysis by using fluorescence. In CyTOF, we instead of fluorescence, we add metal ions are tagged to the antibodies, and these when ionized, so it combines the principle of spectrometry as well as cytometry. So it's a cytometry by time of flight, meaning each metal ions are tagged to various antibodies, and these when it gets ionized, and the plasma so based on the mass, you can separate out what antibody it was tagged to. So that's how. So one advantage is in conventional flow cytometry, you have a limitation of say maybe 15 markers at a time. In a CyTOF system, you can go up to like 80 or 100 different markers and detection channels. So it's from the company called Fluidigm, or now it's called as Standard Biotools, which is you have I think in India there are 4 or 5 institutes where you have these instruments in all over India. So it's quite expensive instrument, but they have an office in India, and they do hire people with a background in cell analysis. It's so this is an example of the CD markers that I told you. So in a conventional flow cytometry, you may be able to just check, look at like say a set of 10 antibodies at a time. But in a mass cytometer panel, you can go up to 30 or 80 markers at a time. So all of these markers are characteristic of some kind of pathogenicity, or you have like say for leukemia, lymphoma, you have specific panels. So you can just run these panels and you get to know what markers are there. So in a conventional flow cytometer, you would have needed say 1, 2, 3, 4, 5 different tubes to run. On a mass cytometer, you can run all a single tube and get all of these markers analyzed. Now, moving forward, we have something called as, so we looked at conventional flow cytometer, we looked at a mass cytometer. The next technology that we have is something called as spectral flow cytometer. So so how it is different from a conventional flow cytometer is, in conventional flow cytometer, we have a limitation of we cannot have too many dichroic mirrors and detectors, because as you keep on increasing the size of the flow cytometer will also go on increasing. So usually you will find 4 to 5 lasers and say 20 detectors. But in a spectral flow cytometer, you can go up to 60 or 80 detectors. So the one of the popular spectral flow cytometer company is Biotech, and so in clinical research where you want to look at say 50 parameters or 60 parameters from a single sample, this is something people go for. So what we talked about was cell analysis. Now, what if you want to sort these cells, meaning you analyze the cells, um, but you now want to get back those cells of your interest. Say, look, in this picture, you have like you have green cells and blue cells, let's say. Now you want to separate the green cells into one tube and blue cells into one tube. So once it passes through the laser, it the detectors will know what kind of marker it has. So if it understands that it is a green, say it's a green cell, the time that it accurately calculates the time that it will take to travel from here to here there's a charge disposition place. So it accurately calculates the time it will take to move from this laser interrogation point to the charging point, and it will give a charge such that it will move. So if it is a say if it is a green cell, it will give a negative charge so that it moves towards the positive plate, and it eventually goes into the tube that collects the green tube, and vice versa for the blue cells. So this is called as fluorescent activated cell sorting, or commonly called as FACS. A popular company that produces such instrument is BD Biosciences, which has its office all over India, and they it's a wonderful place to work for as well. Another company that brings you cell sorter is Beckman Coulter. So what you had here is something that fits into a laminar flow hood for aseptic cell sorting, and when you want to, maybe you are working with some virus particles and all, you need to have safety cabinet. You have benchtop cell sorters as well, which again works on the same principle but in a very compact size. The next one that we can talk about is it's called multiplex suspension array system. It's brought about by a company called Biorad. So basically, here you have a 96 well plate or a 384 well plate. Each uh, wells has beads that are coated in different concentration. And say, for example, you want to analyze some cytokines, and you have you have kits that can do 48 cytokine analysis, meaning so each concentration of the bead will represent a cytokine, and you just have to put in your sample, and you can do duplicates or triplicates or have multiple samples run at once, and based on the concentration difference, it will tell you which cytokine is present and which cytokine is absent. So it's again, so why it is called high throughput is because we are not doing sample analysis one by one. You are able to do 96 well plates or 384 well plates, meaning you can literally do 96 samples at a time, and probably in a half an hour time the entire plate would be analyzed, compared to what you would be doing. So imagine, this is also the underlying principle is again flow cytometry, but in flow cytometer we are running one sample at a time. And here you are able to, and here you are able to run a 96 well plate or a 384 well plate with say up to 40 or 50 cytokines or analytes being analyzed from each well. This is called high content imaging. So imagine you have a microscope. So in microscope, you do the analysis one by one, right? I mean, one person is sitting there and analyzing slide to slide. Or if you have a 96 well plate, you do so you have to do it one well at a time. So this is a fully automated system where you can load all the 96 well plates. You can stack up your 96 well plates and then tell the instrument to go from one well to another and come finish. Say if you have like 5 plates, 5 into 96 number of wells, you can do the imaging. You can do various time point imaging, meaning, okay, if every 5 hours or every 6 hours or every 12-hour time points, if you want to do an imaging, that's possible. It is climate controlled, meaning if you have cells or culture that requires a particular amount of oxygen or CO2 or temperature, that is possible. The chamber is climate controlled or environment controlled, and it also has the option for injectors, meaning if you are doing some calcium efflux assays or in your experiment or an assay, if you want to add some reagent in between and you want to check how the treatment has affected your cells, that's also possible. So it comes with fluidics, it comes with the environment control, and it comes with an imaging capability where you can do confocal imaging as well, and 3D for 3D assays, etc. So it's called high content imaging or high throughput screening imaging systems. It's brought about by a company called Molecular Devices. Again, you will find it by a place, say for example, Rajiv Gandhi Center for Biotechnology in Trivandrum has one of these instruments, along with they have a very wonderful imaging facility and flow cytometry facility as well. So speaking about microscopes, one of the common applications is for slide imaging. Again, we do imagine doing a single slide imaging. Now, we have hospitals where per day, uh, where they do tissue section slides of like say 600 or 1000 slides per day. Now, it's not literally possible for one person, or say even if you hire 10 people to keep on scanning these slides and finish the whole thing in one day. So this is a whole slide scanning system where you can load 1000 slides at a time. So it's from the company called 3D Histech. They have a, single slide scanner, a 12 slide scanner, a 250 slide scanner, 480 slide scanner, and what you see here is a 1000 slide scanner, meaning these are the capacity of slides that you can load at a time. And it's a fully automated system, meaning an robotic arm will come and take each slide from its rack, take it to the put it under the objective lens, scan it one by one, create the images transferred to the server, where the pathologists or the researchers can view these scanned systems from their laptops at their office. So mostly these are implemented at hospitals where the pathologists um, uh, or maybe probably the researchers are sitting at different places, and they can do teleconsultation and have a give out a report um, um, about the case that they are reviewing. another technology that I would like to talk about is a dry reagent technology. This is from Beckman Coulter, where so if you remember, I told you about some CD markers before, right? Say 3 or 4, or say suppose you have 10 CD markers. Usually these comes in 10 different vials, for each vial for each CD marker. So if you want to add if you want to check 10 markers, you have to literally do 10 times pipetting into a tube like this, and then you add your blood or PBMC or cells into it. You incubate it and then lyse, and then you run it on a flow cytometer to check what markers are there. So, for example, when I was at Amrita Institute of Medical Science, I was at a leukemia and lymphoma lab where, for a typical leukemia or lymphoma panel, you will be running around you will have to run at least 10 or 20 markers to, or maybe probably 30 markers to come up with a diagnosis. So that means I was pipetting 30 different, so I will have 30 different small vials in front of me, and I will pipette all these into one tube. So the chance of making mistakes is higher. So this particular technology, what it does is it has all of these markers dried down at the bottom of the tube. So what you see here, a slight reddish tinge, is all the dried down antibodies, and they have already done this validation and optimization, titration, everything for that, so that we don't have to do, or a researcher don't have to do this again. So these are pre-checked or validated panels where you just have to buy this single tube, and you have to add your sample, then your buffer, and you are ready to run. So it saves a lot of time. Chance of making errors is less. You can, so usually for, say, you must have heard of clinical trials, or for so vaccine development and all of these. So typically you will have to do 300, 900, or probably thousands of samples for validation. So imagine each tube. So suppose each patient or each specimen will have, you will be running 2 tubes or 3 tubes, like duplicates or triplicates. Imagine you will have to add 10 or 20 antibodies one by one, compared to you just take this one tube, and you are ready to use. So it's a very popular one. It's used a lot in researchers nowadays. And one of the best thing is it's made in Bangalore. So so all of the kits that we get will has a made in India branding in it, and that's something I am like I tell people, I am very happy to tell people here that it's made in India, and it's a technology that people love, because how easy and it is to use, as well as how and the results are always really good. So all they have to do is arrange the samples from various places, and a 300 patient publication is about to come out soon from one of our customers here in Qatar, where it's a research center where they did a 300 patient sample using this particular tube. So it's this is an example of a panel that these so it's called Duraclone. These tubes are called Duraclone. And so, for example, if they want to run a mesenchymal panel, so all of these markers are already there in the tube. So otherwise, you would have to have all of these markers, individual vials, and you will be, pipetting to each one to one tube, and then you have to run that. Imagine having to do that 300 times for 300 patients. So if you have this tube, you just have to buy a 300 set of tubes, and you just have to put the samples in 300, and you are ready to run your samples. So I would like to tell about like what we do here in our company. Like we represent these manufacturers. So we are kind of turnkey solution providers where we are just not selling these um, products, but we kind of make a workflow out of it and help the lab set up the whole workflow. So, for example, if it's a library preparation or a sequencing workflow, we are partners with company called Twist, Beckman Coulter, Illumina. Warsom Clinical is a bioinformatics company, and IDTs are primers for library prep. And so we provide the whole solution so that the researcher can have so they can buy the whole thing from one vendor, and we have people who go and support them in their application and workflows, troubleshooting, set up the experiment, and then they do the whole process. So, and another example is like what you see at the bottom, like we have labwares for sample preparation, for high content screening and analysis. So, we kind of provide an end-to-end solution for the complete workflow. One of the most what to say enjoyable things that we get to do is do when we go around supporting in their panel designing, in experiment preparation, or designing the experiments. We do demos, we have conferences, and we do post installation trainings for the customers. And it's something it's so I don't see many of biotechnology people moving into this category. I don't know, probably they are not really aware of these companies, or mostly it's biomedical people as and the molecular biology, biochemistry, and those people that come into I mean, most of my colleagues or whoever I see are from in that. But this is something that everyone in biotechnology can look into. It's a career option that is very interesting, as well as what to say, like enjoyable. So that was I think that was it. If you can connect with me on LinkedIn, and if you have any questions, feel free to reach out to me.
Participants can post their questions in Q&A or as an webinar chat box. There is one question, how to find workshop and conferences in biosciences or medical science industries.
Yeah, actually, I would recommend. So if you visit all of these companies that I have mentioned, and there will be newsletters, and you can follow their LinkedIn page, of course, as well as there will be newsletters where they constantly tell you the webinars that's happening. So all of these webinars are will be talking about their technology and the principles. So it's a very nice way you can learn about these things. And I would recommend, when you have say you during your semester breaks or your industrial visits, you can visit these either, if not the companies directly, you can visit the research institutes that have many of these systems. So for example, you can Rajiv Gandhi Center for Biotechnology is a place where a lot of people come for industrial visits and their semester, what to say, the project for doing their projects and all. So it's a nice place to get exposure to these technologies, and you will get an idea like what. So whatever I have mentioned in these are not like very high-end technologies that you don't find around. These are something that I have used, each one of them, and you will find it in the research institutes around. So, for example, at Rajiv Gandhi, in Trivandrum alone, if you'll find at least 10 high-end flow cytometers. You have Amrita Institute of Medical Science. Again, you have they have a research facility where people come. So when I was working there, people used to come there for as part of visits and to visit the place and for trainings and stuff. So yeah, reach out to these centers and it will definitely be a good experience.
Okay, sir. Then there is a question that what kind of job roles are offered to biotechnologies in the companies which you have mentioned?
Yeah. So see, these are all basic instruments that you will find in research centers, right? So the first thing that in order to get a job in these companies would be to you will need to have an experience in any of these instruments. So the best way is so as a biotechnologist, we are mostly very much focused on either lab work or we may be talking about pharma and production in that area. So I would recommend get an experience, at least a 6 months or a 1 year experience in a research lab. So you will get to know, okay, what are the actual things that are instruments that are being used, and you'll get to know from the seniors there what are the opportunities that they are getting in terms of PhD or other stuff. So once you have and you will have to make your way some way to get experience on these systems. So for example, I started at RGCB. So there we were handling confocal microscopes, flow cytometer, and a sequencing facility. So kind of realized, okay, clinical flow cytometry is a place where not many, and it's something, it was something not very commonly you can get experience on. So I made a deliberate attempt to move to Amrita Institute of Medical Science, where um, you get an experience in clinical flow cytometry. So it's basically a clinical lab where you have actual patients coming in for leukemia and lymphoma analysis. So that is the only way you can get experience. And so if you by end of 3 years or 4 years, you'll be good to. So once you have confidence in any of these any instrument technology, you will be able to apply for, and they usually will not, they need only like 4 or 5 years or 3 or 4 years of experience. uh Main thing is, if you are confident on the instrument by end of 1 year or 2 years, that's fine. So and for example, say BD Biosciences with a 4 years of experience in flow cytometry would give you. So this is from 2016, I'm 16 or 17, I'm telling. They offer somewhere around 70,000 per month for that. And somebody with say 7 or 8 years, Beckman Coulter or BD offers around 1 lakh per month, and it goes on. So somebody with 10 to 12 years of experience would get so by that time you would be more moving more into the marketing, because technical is always your base. And you see every company, not just company, everywhere, the commercial aspect would come into play. So eventually, the higher you go, it's more commercial you will get. But again, you will be seen as a technical person, because that's the base. So as you move more into commercial or marketing, um, the package also will go up. So it's a very a really good area that people should look into, I would say. Yeah.
Thank you, sir. Then there is one question that are there any Bangalore-based hands-on courses on high throughput screening and cytometry?
There are a lot actually in Bangalore. There is NCBS. So NCBS is where you can approach and ask them if they have regular courses, that I'm sure, because I think once we visited some probably 8 years back or something. So they have NCBS has courses, regular courses. IISCA also has courses. Rajiv Gandhi Center for Biotechnology has a regular flow cytometry course every month. They have every month. Yeah.
Okay, then there is one more question. Can you mention any job opportunities for microbiology freshers in the companies that you have mentioned?
They uh, for freshers in the company, it won't be there. I'll be honest, because so for a fresher, the best place to go and approach to get experience is a lab, because when we graduate after bachelor's or master's, all we know is some theoretical part or the basic lab work that we have done. But it will be good. You don't have to work for like 3 or 4 years in a lab to get experience. You work 6 months or 1 year, you'll get an idea what you're doing. Then you can start approaching these companies. There are a lot of so microbiology. So it doesn't mean only these companies will take from all kind of background. So there will be a microbiologist. Anybody can do this imaging or flow cytometry or any of these things. It's something that anybody can do. Yeah. So the main as thing would be to get an experience, a lab experience, so that you can go and apply to one of these companies. And you can approach any research. You have lots of research institutes where you can apply for JRF or research assistant positions. They frequently take people who have completed masters. Yeah.
Okay, thank you, sir. Are there any more questions? They can post it in chat box. Okay, so there is one more question that can a biochemistry student join cell imaging?
Sorry, what?
Cell imaging. Can a biochemist join cell imaging?
Yeah, anyone can. There is no restriction who to. I mean, so cell imaging. So how I see it is, it's like taking photographs. So you don't have to go and learn photography to become I mean, if you anybody can do it. Only thing is, once you start doing it, you do a lot of that and you become good at it. So it's something anybody can do.
Okay, then there is one more question asking in what type of lab or which kind of lab they should work after their MSc or during their MSc to fetch a good job in core domain.
In core domain,
Yes, sir.
um so see, suppose if you are, so what you mean core domain? Like if you are a biotechnologist, you see, actually biotechnology is kind of wide. So you have that flexibility to move into any lab, because you kind of learn almost every subject in life science in under biotechnology. So based on your interest, you can move into the kind of lab that does. So if it's a cancer lab or a stem cell lab. So, or if it is a microbiology or biochemistry. Uh, so whichever is your area of interest, move into that kind of labs. Check which research institute or which place offers that kind of experience, and you move into that lab. I would don't wait to or don't get comfortable with a particular place or position. um, I mean, don't think of getting comfortable at least um, until the age of 45, I would say.
And they also want to know how to approach this biotech companies through LinkedIn or mail, or directly contact. How can we directly contact HR or the authoritative person in those
Yeah,
companies?
so you can so all of these companies would put their job opportunities on LinkedIn. And so what you can do is just search for application specialist or product specialist on LinkedIn, and you type, say, product specialist. Say, suppose you know some centrifuges. That's the that's one instrument that suppose, or PCR machines. You just type product specialist PCR, and you will see the job opportunities that is there, that is open. You go and look at what are the things that they have put it in the job description. So you will understand, okay, these are the things that you will need to learn in order to apply for such position. So say you have graduated and you are in a lab and you want to apply for a particular post. So you can try to concentrate, or like try to get those skills or those experience. And yeah, you can directly approach the HR, and they will give you a call. Or these company people frequently come to visit your labs. So so once you feel confident on any of these instrument, you can tell them, see, I would be interested. So for to me, I get a lot of questions from technologists and technicians in various labs, telling, okay, if there are opportunities, let them know, because they would like to transition from the lab to a commercial position. So it's some yeah, you can do the same thing. You can network through LinkedIn. You can reach out to them, but just make sure you should be confident on that particular instrument. So like it's like you can't unless you know to drive a car, you cannot go and tell somebody that you want to drive a car. Like um, you need to you get so once you are confident about whichever instrument, it doesn't have to be a very high end instrument, because even if it is a centrifuge or a PCR instrument, you need a product specialist for that company to support those instruments. So for example, Eppendorf or Biorad or any centrifuge company that there will be a product specialist for their centrifuge to market, or to technically support centrifuges, because it's there are a lot of things a centrifuge can do. There are various applications a centrifuge has to can perform. So we for almost every product has a product specialist. A product specialist is somebody who will understand uh, A to Z of that particular product, and will support the customer in how to use it, and or all of the or in their applications.
Okay, sir. Then there is one more question saying that, one minute. Are there any institute or laboratories that you can recommend for doing internship in biomedical sciences?
Biomedical sciences.
Yes, sir.
So biomedical sciences means you okay. I mean, if the two places that I definitely know, two or three places I would say is you can approach Rajiv Gandhi Center for Biotechnology or Amruta Institute of Medical Science in Kochi or NCBS in Bangalore. IISc also offers these internships.
Okay. And they're asking, are there any good opportunities for molecular biology field in India?
So molecular biology would mean so all so we just had this COVID and the pandemic, and now we are in a stage where they are looking, most of the research is going on in precision medicine and clinical genomics. So this is where molecular biologists um, can look into. So a lot of companies are there that, so I would say there is a company called Premas. I don't know if you guys are aware, Premas Life Science. So Premas is a company similar to what I told you about. It's in India, and so they support. They are channel partners for a lot of companies, and they create end-to-end workflow solutions for various research institutes, and um, they hire a lot of people from molecular biology background.
Okay, sir. And they are asking for the company names in cell image. Can you mention them again? Some
In
company name
in what
in cell imaging.
companies in cell imaging? Um, so any all of the microscope companies, like you can start with Nikon, Olympus, Zeiss, Leica. Leica and all has office in Bangalore. They have a incubation center at NCBS itself, because the engineer who used to come when I was at RGCB, the engineer who used to come to for installation and training us was used to work at NCBS. So yeah, Leica, Zeiss, Olympus, Nikon, molecular devices. So these are the imaging companies.
And they are asking, are there any skills that they should scale up to get a job in this field as a fresher?
Um, you should just the only skill would be to get experience in one of these instruments. So as I said, the best way is get an exposure in one of the research centers as a so at least you will get to know like what's happening uh, at in a lab. So yeah.
Okay, sir. Are there any more questions? So they are asking, how much is the average pay for instrument handling profile?
Instrument handling, what?
File.
In,
Not sure. Can you rephrase your question, Prachi, if possible?
is the question here, or I don't see the question?
In Q&A, sir, there will be one.
How much is average pay for instrument handling profile? I know only, okay, 10 years back it was around 20,000 if you are a fresher. But I believe now it's 30 or 35. Um, yeah.
Are there any more questions?
So for jobs for MSc Biomedical Science, so see, basically the first idea would be to get exposure or experience in some kind of instrument handling. So even if it is biomedical science, biotechnology, um, don't constrict think that you can go only in one direction or anything. You can always um, learn additional subjects. So there are a lot of biomedical engineers who later on are have moved into technical roles, and a lot of biotechnology guys who moved into biomedical engineering job roles as well. The best would be to get an internship. Then you will get an idea, okay, these are the labs or places where you can work, and then you can move into techno commercial roles. So all of these are techno commercial roles, meaning eventually you are a part of a company that sells an instrument. So it's like uh, for example, if you like cars and you are a fan of say a Ford company or Volkswagen, you would not be you would need to know the cars and how the principle of working and stuff, but eventually you will end up in a company that's eventually selling cars, right? So yeah.
So there is one more question, that are there any internship available now, online or offline, with stipend?
That I'm not sure if something you will need to check their websites. You can check the website of NCBS and RGCB, Rajiv Gandhi Center for Biotechnology, and NCBS. They would have definitely.
And
Or you don't have. I mean, see, they sometimes take it as a job opening also. It will come because what we I got into was it was a position for biotech engineer to handle the instruments, and so they have frequent openings because it's under central government, and they have frequent openings. So you just keep following their website. They call for research assistants, research associates, JRFs.
there is one more question, sir. That can you suggest some laboratories to carry out the research work related to cell lines and molecular techniques?
Uh, to recommend, see, I don't know if you'll have to speak with the PIs there and see if they will be interested in collaborating or to do some of the techniques. So if you are looking at private labs, I can't recall many of the names, but a lot of private labs are there in Bangalore and Chennai, etc. So yeah, you can approach them. Or if it's the research institutes, again, I can tell you about only just there is IISER in Trivandrum, apart from RGCB, which is also then there is so in Trivandrum there are like 4 or 5 good research institutes. There is tropical uh, tea uh, tea tropical botanical research institute, something like that. So yeah, you can check those research institutes.
Are there any more questions?
For cell imaging, no entrance exam is there, but I saw one of the questions. So
Any more questions?
yeah, TBGRI. Yes, thank you. Because people used to come from there for cell cycle and ploidy analysis on flow cytometers when I was at RGCB. So they used to bring samples for running.
Okay, I guess that's, if there are no more questions, I think there is one more question. So they're asking if any hospital provides internship for biotechnologists.
There are so any hospital that has a research institute also attached to it, they provide. So there are lot in Bangalore. Another I know is Amrita Institute of Medical Science, MIMS in Calicut. Then there is SCT in Trivandrum.
Okay, sir. Are there any more questions? I'm writing the company names that sir has given to everyone so that they can have access to it. Apart from that, is anyone having any questions? They can post it. Yeah, I guess that's it, sir. Okay, sir. And we'll proceed to finally close the webinar session for today. So I would like to thank you for your engaging and interactive session. We believe that our participant would be having deep insight into whatever you said today, and they will be definitely benefiting from it, and will be going through whatever institute that you have given. They will go for it, and they will search for internship and whatever suggestions that you have given to them. Thank you so much for today's talk, sir.
Thank you everyone for attending and for a lovely interactive session.
And for the participants, the feedback link will be provided shortly along with the recording. So you can give your attendance over there. Thank you so much, everyone, for the session.